Okay. I know I'm in between you and lunch, so I will stay on time here. A tour of DANDI datasets and search tools. All right, so now you know about NWB and DANDI, and you know how to read NWB files, but how do you find the ones that are most relevant for your science? We already talked about there's tons of datasets, there are different species, there are different recording modalities, lots of different data types. We talked about the highlights, such as microns, IBL, Allen Institute. You'll hear more about those later today. And hundreds more. Okay, so what about the hundreds more? How do we actually navigate across them? Well, the easiest way is just using the DANDI search bar. So, I mean, this is pretty straightforward, but if you go to dandiarchive.org, you can just search for like neuropixels. 41 results found. So, I mean, that might just work for you right there, right? There's no need to go into more complex things. I mean, you could look for like hippocamp. All right, so I'll do hippocampus. And there's 63, but if I just type hippocamp, there's 95 because a lot of them say hippocampal. So that's the points to the downside of the search, right? Because it's a literal string match. So you kind of have to know the tricks. And if you use a space, it's like an AND operation. So you could do like that and mouse. There you go, 34 data sets. But you can also like rhesus, does that work? Yeah, okay, so 2 data sets that specifically record for the hippocampus in rhesus macaques. So, you know, it's not that sophisticated, but it might be sophisticated enough for you. So there you go. There are also much more advanced ways of doing this through the Python API, which I will, I'll just show you I'm not going to walk through these in a detailed way right now. I'll just kind of show you what they look like. But essentially, DANDI has sophisticated asset and data set level metadata, and you can use the Python library to query them. So if you pip install the DANDI Python library and you instantiate a client, this is all free. You can do this without even a DANDI account. Then you can search for things like searching for data where NWB is in the data standards. And you can look for data sets where mouse is in the species name. Now you do kind of have to know the structure here. You can also do more complex things. So you could look for data set, like if you look like within data set 5 for assets where the species is a mouse and the sex is female. So to do these, you really have to know the metadata structure of the assets and the data sets on DANDI, which is I've printed out here an example. There's a lot here. So there's metadata about the anatomical structures being accessed, keywords about the contributors and the roles of those contributors and the affiliations. So you could query based on that. About the species and the approach, the data standards, the number of bytes, files, subjects, what variables were measured, measurement techniques, the license of the data. So there's lots of metadata that you can query on using this Python API. It'll just, like the program I have here just iterates through every DANDI set and just like runs this filter. And that takes only a few minutes. So you don't have to do anything more sophisticated than that. For those of you who want to use these more advanced tools, I'm happy to like work with you to figure out how to create the query that would be right for you. But I think the other tools that I'm about to show might be more relevant. So another tool we have is semantic search. So this is a text-based search, but it's not doing a strict string matching. It's actually using LLMs to try to do a semantic matching. So this is part of an approach that's called RAG. So essentially we take the metadata of every DANDI set and we pipe it through an LLM and we get a vector that represents the semantic meaning of each DANDI set based on all of that metadata that you just saw. And then later when somebody types in a search query, we can run it through the same LLM, get a vector, and then we can look for the nearest vectors to the ones that we stored. So this is great because it gets around the like hippocampus problem, right? If you say hippocampus and a data set says hippocampal, that'll be very close in semantic space. So it gets around like other things too. So let me show you. It's in this neurosift.app thing. So there's a data set that we published recently on DANDI that was from NASA. Actually, it was rats doing a 3D maze in outer space. And I'm going to search for it. So if I go here and I type NASA, there it is, 3-dimensional spatial navigation of hippocampal neurons during space flight. Okay, but let's just say I didn't know it was NASA and I type astronaut. That's not going to come up because the word astronaut just didn't appear anywhere in the metadata of that data set. But if I go over here to semantic search and I type astronaut, here we go. 3-dimensional spatial selectivity of hippocampal neurons during spaceflight, and it talks about the space shuttle and it talks about NASA, but it never says the word astronaut. So this is another way to do search. Now, pros and cons. It will do semantic matching, but you wouldn't want to search something like, give me the top 5 data sets. Like, it doesn't know how to do that. It's not really an agent. It just does semantic matching. Okay, so one of the biggest pieces of feedback that we got from last year is that people wanted to do better queries over brain area. Now, this is something that's actually quite difficult to do, to get the metadata standardized across all of these different labs to query brain area, but we've made a lot of progress here in a tool called the DANDI Atlas. So here we have a collection of all of the data sets where we've indexed the brain area that are recorded in each of these experiments. We have a bunch of different atlases and different species. We've got the Allen CCF for mouse. We have 3 different macaque atlases and 1 rat. We have way more data for mouse, so I'll click that and show you what that looks like. So this opens a 3-dimensional atlas where you can navigate the brain and find data sets that are record from a given region. So I'll just navigate in through the hierarchy. And these numbers here indicate how many data sets within this parent region have recorded from a region within this parent region. And the red is, they label this region specifically. So I'm going to look for visual areas. We've got 24 in visual areas. Okay, I'll just click the visual areas. And then on the right here, you see the data sets that, let me enlarge this a little bit, that have recorded from this area. So I'll click here, this Allen Institute visual coding, and I'll just click on the first subject, and you can actually see where the neuropixel probes were. So this red dot indicates that we have actual 3D localized positions of the electrodes, many of which are neuropixel probes. So here is one session from this subject, and you can see exactly where these electrodes have been localized within the Allen Institute CCF. So we have this for dozens of different data sets. I actually was really amazed when I saw this, like how much coverage we have of all these different brain areas. And so if you have a specific brain area that you're interested in, go ahead and try to filter on that and see if what data sets record from there that might be of interest. And there's a lot of tools for interacting with this. These give you different views of the brain. You can play with the transparency of the regions and the electrodes. You can make certain brain areas disappear to get a better view of others. Okay. If you click on this arrow, it'll bring you to the actual data page for this subject. You can also open this Dandiset page. Let me just show you really quickly another atlas. I'll show you the D9 atlas. for rhesus macaques. And we've got only one data set that actually has electrode positions. And if I click on a subject, okay, then we can see where these recording sites were. I'll just zoom in here. And then another subject, here are the recording sites. So you can see maybe if you really care about anatomical localization within a larger brain area, dorsal versus ventral area or something like that, this could allow you to answer those types of questions. This is the first data set that we have that has proper anatomical localization on a rhesus macaque in DANDI. So we're working on more, but it turns out that registering locations to a common atlas can be really tricky, and especially for species other than mice, where you have less standardization of the atlases. But yeah, this is a fairly new tool, so please check it out. Just play with it and see if you can identify data sets that are relevant for you, and I'd love to hear your feedback on that. Another tool that is really new, as in I made it 2 days ago with Fable, but I think I realized that this would be really useful for us, so I'm curious what you guys think. So this is a map of the semantics of all the metadata. So basically all the metadata for each DANDI set is passed into an LLM represented as a vector, and then we do dimensionality reduction and projecting it onto a three-dimensional space. And so these are all the data sets on DANDI, and the spacing and the coloring indicates similarity between data sets. So this will really give you an idea of all the data sets on DANDI, which I think is really hard to do otherwise. And the clustering here really represents different types of data. So if we look at this is a pretty pronounced cluster here. These are all ultrasound, transcranial ultrasound data sets. This cluster here is all C. elegans neuroimaging. All the way out here, we've got fish behavioral data. Here we've got zebrafish behavior plus neural recordings. And let's see, out in this big cluster over here, we have all patch clamp intracellular electrophysiology data. So if you find one data set that's kind of similar to something you're looking for, maybe identify that data set here on this atlas and then look for data sets that are nearby. You might find an even better data set. Okay. Any questions so far? All right, great. Okay, I want to revisit Neurosift because we put a lot of work into this in collaboration with Jeremy Magland at the Flatiron Institute, and that has a lot of capabilities. One thing that hasn't been shown yet is the PSTH, right? We haven't done that yet. All right, so I'll do that. And I'll start here at the IBL brain-wide map data set, which we'll hear more about later today, but I'll just give you a sneak preview. So I'm going to go into Files, identify the first subject, open up. Notice this is a 71 gigabyte file, but that's not a problem because Neurosift streams the data and won't be accessing the vast majority of that. So I'll open it up in Neurosift. And I need...
Split across raw and process now.
Oh, did I open the wrong one? Maybe I did. Yeah, I want PSTH. Okay, so this is streaming the data live from the archive and generating PSTHs of each neuron. So here, each row is a trial, and these dots are the spiking of this neuron. And this is just an average across all of those trials. And we can align, let me just enlarge this a little bit. Well, align by auditory cue time? No. Gabor stimulus onset time. And let's do wheel movement onset time. And then we can group these by the Gabor stimulus contrast. Okay, so this is one neuron looking at its response to different stimulus contrasts of the Gabor. This is aligning to the stimulus onset and aligning to the wheel movement onset. And you can scroll down and see the response of all these different neurons. You can see nice graded responses for many different neurons these are clearly very aligned to the stimulus onset and increasing. But that's not always the case. If we keep on scrolling down, in some neurons, we see a decrease in activity with stimulus onset, right? So you can really start to get a sense for all of these, the activity patterns of the different neurons without ever even doing any programming, just by looking at this. And yeah, I mean, I could look at this stuff all day. I think it's really fascinating. Like, what are the stimulus aligned to? Do they increase or decrease? How do they change based on the parameters of the task? I mean, there's so many different questions you can get at right here. And yeah, so this visualization was not designed specifically for IBL, right? This will work for any data set that has spiking units and a trials table with different categorical variables. So dozens of different data sets on the archive. Well, maybe it won't look as good as this, but you'll be able to run these features. Any questions so far? I know I'm going a little fast, but I think you guys can handle it. Okay, maybe too fast? I don't know. Too fast? Just right? We'll see. Okay, Ryan already walked through this data set from the Moser lab, but I wanted to take it to the next level. So let's say you have identified this data set. Ryan showed, okay, we'll visualize this, and we'll visualize all three of these streams. Okay, and you say, yeah, you take a look at the data, you say, yeah, this is exactly the kind of data that I want to analyze. What now? I mean, you're not going to take a screenshot of this and put it into paper, right? Like, how do you actually go from here to analyses in your notebook? And that's where I would recommend looking at Python usage. So as Oliver was going through the last tutorial, he showed you exactly, really in detail how to access each data element. But you might be wondering, well, how is this going to look in different data sets? They all have different types of data. They have different types of stimuli and responses. Some are electrophysiology, some are calcium imaging. So this answers that question. Basically, it's an automatically generated script that shows you how to access and stream any data element on any NWB file on DANDI. So you can just copy and paste this in. So I'm going to take this, I'm going to copy it, and I have a notebook that I just started. Maybe I don't. Oh yeah, over here. And it's gone. Okay. Let's start it back up. All right. So I'm going to, I'll show you the installation cell, which I'm not going to need, but all of the tools we're going to use here are PyNWB, LinD, Pineapple, and Matplotlib. So just make sure all of those are in wherever your environment is. And then let's go back to here, copy, and paste. Okay, so now note this is, it already has the location on S3 of where that asset is. It sets up this it's not using REM-file, it's using LinD, which you'll learn about later. It's another way to stream the data. But at the end of the day, you just have this NWB file object that's streaming the data from the archive. And so if I just type in NWB, you get this interactive object that you can use to explore. So here, units, columns. It's just streaming the data and showing you the metadata here. I mean, you could do NWB file dot session start time. Okay, there you go. That data has come in directly from the archive. And you can do, let's do something a little bit more interesting. Oh. It's just one-dimensional. Okay, there you go. That's data downloaded from the archive, right, that you can analyze. It's that simple. Okay, now I want to show you a demo of Pineapple. I'm going to steal Guillaume's thunder a little bit. He's going to do a whole session showing you exactly how to use this, but I just want to show you one demo, maybe to get you excited about it for tomorrow. But essentially, Pineapple defines objects like time series, intervals, timestamps, and then methods on those objects, which can be used to construct common neurophysiology analyses and make it really fast to do so. And it interacts with, it can easily ingest data in NWB from DANDI. So I will do import Pineapple as nap and import matplotlib.pyplot as plt. Okay, then I'll do nap NWB equals nap.nwb file. So this is the first step, which is ingesting that NWB into Pineapple, where Pineapple looks at this NWB object and extracts the streams that it can represent. So it says, oh, okay, I found units, I found electrical series, I found a spatial series, and I found another electrical series. So these are now Pineapple objects that we can compute over. So Pineapple also defines a lot of convenient analysis functions. So let's do rate maps equals nap.compute tuning curves, nap NWB units, nap NWB and we'll say 30. Okay, so this is computing rate maps. It's already done. So for x in rate maps, let's do plt.figure, plt.imShow x, plt.color bar, label equals firing rates hertz. And then there you go. Grid cells in the medial entorhinal cortex. So there's your Nobel Prize speed run. It's really that simple to pull data to find a data set using all these different search tools, right? Stream the data using the copy and pasted code from Neurosift, and then load it into Pineapple, and then you can do rate maps, and then Guillaume will show you tomorrow a dozen other tools you can use. And you can just throw it into your own analysis libraries. You know, you don't have to use Pineapple, but it does provide a lot of convenient functions. Okay. All right, I showed that, and that was in case it didn't work. Okay, notebooks. So this is one example of an analysis of one data set. We have, over the years that DANDI has been here, we've been encouraging people to submit notebooks demonstrating analyses. And a lot of these are reproducing key figures from the papers, right? And this, if you're using these notebooks, allows you to really hit the ground running and build upon great visualizations, great analyses that have already been created. So we have a GitHub repository here, which is a collection of all of these notebooks, which is rendered here at notebooks.dandiarchive.org. And we have maybe about 40 different data sets that have at least one notebook. We test these all on a Google Colab-like environment, and if it runs, then we'll put a link here. The last one we worked on was this, or the last one that's been added. There's a lot that are in embargo still, but the one that is public is this NASA data set. So let's open it. I'll show you opening it in Colab. And yeah, I mean, this is, again, we're using Pineapple here. We're calculating rate maps. I'm showing you how to access some basic metadata. And here's the occupancy map in the maze. And if we keep on scrolling down, here's a raster plot. And here's the pre-calculated rate map versus what we calculated using Pineapple. You know, they did differences in filtering and stuff like that, but generally we have the same results as the scientists who submitted this data set. And, okay. Now, if you go through data sets and you find that there are problems with those data sets, like the RGB being BGR or something like that, or something more severe, if it might happen, there could be key metadata that could be missing or incorrect. There could be raw or processed data that's missing. They could be not necessarily incomplete, but just in a structure where you don't understand it, where things are just given like integer labels and you're like, I don't know what any of this means. The channel to discuss that would be the DANDI help desk in discussions. So if you click this, you'll see there's a label Dandiset issues, where this is our best approach right now. Like, we don't have the specific ticket tracking for specific Dandisets, but this at least allows us to keep track of the issues and contact the authors and get their input and hopefully fix these so that these data sets become more reusable. Okay, I actually do have more time to go over the advanced search, but I wanted to make sure I answered any questions first. So we have 15 minutes left. I was able to plow through this. But yeah, I mean, this is your chance to ask questions about identifying data sets that would be relevant for you. Yeah.
I think I'm nitpicking, but here you use Lindy.
Can you talk into the box?
Sorry.
Thanks.
Hi. What was I saying? Here you use Lindy to access the NWB file, and in the previous tutorial we use REM.
Yeah.
So when would you use one or the other?
Ryan will go over that a little bit, but it's there are lots of ways to access the data, and they all work pretty well. You can use REM file. So Lindy can be more efficient, but it requires that there be an indexed file created already, which is only true for published data sets. So Lindy won't work if the data set is in draft mode and it never has a published version. So in that case, you'd have to use REM file. Is that? It's not guaranteed to exist?
Yeah, it's not guaranteed.
Yeah.
Neurosift indexes DANDI regularly, but it doesn't fetch them all.
Yeah.
So there's not always a Lindy file.
But I think that the automatically generated code will give you the one that it's able to do. So you don't have to worry too much about it. Just copy and paste the automatic. And then after that, those first few lines where it's like whether you're using REM or using Lindy, or actually there are other ways to do it too. Like there's FS-Spec, which would allow you to read it from like a Google Drive, like stream it from a Google Drive if you wanted to. Like there are certain differences in the functionalities of why there are all these different approaches. But at the end of the day, you just have this NWB file object that you can kind of ignore like exactly what streaming layer you're using.
Okay, thanks.
Okay. I'm going to go back then and walk through the metadata searching. Advanced. Okay, let me see. Did we go all the way through the simple? Yeah, basically. All right. I'll just show you one more thing with the advanced metadata searching. So here we're going to set up a DANDI API client, and we're going to iterate over all of the DANDI sets and extract a list of DANDI sets. iterate over all of those and get the raw metadata for each one. And this is going to allow us to filter on this metadata. So first, let's just make sure we're only looking at data sets that are of DANDI. So here is the RRID, which is like a registry of electronic resources. This is the one corresponding to DANDI. So we're filtering all of the data sets based on ones that match this RRID. Okay, now we're just considering data sets on DANDI that are in NWB. And then let's say we wanted to determine something advanced, like we wanted to filter by the average age of subjects in a data set. Then you're going to actually have to do some computing on the values of that data. So here is a function that takes the age and turns it into like a numeric representation. And then we can say, okay, well, let's look at this data set, 398. We'll get all of the assets, iterate over those assets. This TQDM just gives you a progress bar as you go over those assets. Extract the metadata corresponding to each one, get the subject, and then for the subject metadata, if there's an age, then we can extract that age and append it to a list. You might also want to, for instance, count the number of spiking units across all sessions in an experiment. So that's something that is actually going to require you to read the NWB files because it's not metadata that's extracted into asset level metadata. It's just something that will require you to actually stream the NWB file. So here we're going to load in PyNWB, and this is yet another way to stream the data using ROS3. But at the end of the day, you just have the NWB file object, and you can, and it's just the length of the units table is going to tell you the number of sorted units. So then we can get a dictionary of all of the S3 asset URLs and the number of units for that, which will be something like this. And then, you can take the mean, you can take the sum, whatever you want to do. So this is a demonstration of a combination of querying the DANDI asset and DANDI set level metadata, as well as streaming the NWB file and asking questions that actually you can't ask with the metadata, you have to ask with that NWB file, right? So you can actually do just about anything you want with this, right? You could say, oh, well, I want a certain signal to noise, or I want a certain number of units that are qualified as good, or something like that, right? So you can really go fine-grained if you're willing to iteratively stream each of the NWB files. And again, here's just the raw metadata dump for an asset, so you can see the structure of that and all of the different fields that you're able to use, things like genotype and what type of neurodata objects are recorded in that NWB file. Okay. Any questions about that?
So why are there so many streaming?
Well, it's just been many years, and the technology has evolved. So why are there so many? So like FS-Spec allows you to stream not just from raw S3, but like from any remote drive. So if you want to do it from like a Google Drive or something, you'd want to use FS-Spec. The one I just showed here, ROS3, is developed by the HDF5 community, and it really wasn't very good at first. They've really improved it lately, so now it's become a more viable option. And then we've built much more lightweight streaming layers in Python that just get around all of the like complicated stuff going on in HDF5 and just basically say, whenever you do a read request on a file, instead of doing that, just like download that range from the remote source. And I mean, it's a little bit fancier than that because we like want to package range requests together. So we're not making like thousands of tiny requests, but basically that's the idea. And then that issue of making thousands of tiny requests was still kind of a problem. So that's where we developed Lindy, which pre-indexes the file into a JSON. And then, so you don't have to make as many tiny requests and you're just really accessing the file for the heavier data objects. So that's kind of our latest generation of this, of like streaming technologies. But honestly, like depending on your use case, any one of those could work.
So my conclusion from this is that I should first go with Lindy and then.
Yeah, I think Lindy is probably the highest performance. So if it works for you, it's probably the best way to go. Yeah. Yeah, thanks for the question. I didn't realize until just now how confusing it is that every single time we stream data, it's with a different package.
Another nice thing about using the copy-pasted script from Neurosift for your data set of interest, beyond it using Lindy, is that it lists every way to access each of your individual data elements in the file. So there's those are all of the Python commands. You can just look at them and just copy and paste them as you need and adjust. You don't want this row or this array or whatever. But you could also, for those of you who are using AI coding agents, this can be a nice cheat sheet for your AI coding agents to know what's in the file and how do I access it, instead of it trying to guess based on interactively exploring your data. This might save you some tokens.
Yeah.
So I see that sometimes besides the Neurosift option, we can even load the files in the Datasets. There's also sometimes one for the Explorer.
Yeah. So this menu is for external tools. So any tool could be registered to Dandy that allows us to open this and other tools. And actually, if you open up there's some neuroimaging data sets that have completely different set of tools. And these are ones that have been registered that it could be used on this file. Metacell is a company that developed NWB Explorer. I haven't used it in a long time, but they also provide some interactive visualizations. It's not something that I actively work on, but I know that they really leaned into intracellular electrophysiology visualizations, so that might actually be really useful to check it out here. But you can already see some similarities in the way that they're displaying the metadata of the data set, and they're, I guess, pulling in. I'm not as familiar with this, but in the future, we're imagining a dropdown that has a bunch of tools. Anyone who wants to submit a tool. Good question. Thanks.